Bismillahirrahmanirrahim. Assalamu alaikum. Welcome to my YouTube channel, Science for Everyone. Today's video is related to gene set enrichment analysis, and the title of my today's video is that how to prepare data file for gene set enrichment analysis. This video is very much important for you because basically when we are using different softwares and tools to process our data, so they need a special type of data format, and it also need a special data type. That what type of data should be arranged in the rows and columns of that file so that it should be accepted by the software or the package. Okay, so this is very much important for data analysis. So for gene set enrichment analysis, we use a specific data file which is basically consist of rows and columns that consist of gene symbols and log FC values. Okay, so in a minute I will show you the file and how does it look like. But first, I should give you a brief introduction about it. What is basically a gene set enrichment analysis? I will not go into the detail of the gene set enrichment analysis, but just a quick overview that basically molecular profile data is our input data, and the gene set database is basically the database that consists of reference genes with specific log FC values. So our data is compared with the gene set database, and after that, we run the GSEA analysis and enriched sets are produced. What does this mean? Basically, if I explain it in a very simple way, so molecular data is our input data. Suppose the input data is our cancer patient data, which consists of gene A, B, C, D with a specific log FC value. Okay. Now, what is gene set database? Gene set database basically consists of experimental derived data of normal individuals, which consists of genes that have specific log FC value or specific expression. So basically, it consists of genes sets that have specific expression values, and it is related to different pathways. Okay, so for example, just we want to take a gene set or gene set of glycolysis pathway. For example, there are 4 genes A, B, C, D, and a product is formed. For example, it is a glycolysis pathway, and we know that the expression of gene A, B, C, D in the glycolysis pathway is experimentally derived, and for example, it is 1. Okay, for all the genes. Now, what does happen is that when we input our data, okay, so it is compared with all the gene sets present in that database. So it could be different pathways or it could be different other biological processes. Okay, so what does it do? That, for example, our database consists of a glycolysis pathway gene set. Okay, so our input file is compared with it, and after that, suppose we say that all the genes that are present in our input file is also present in the glycolysis pathway. So basically, it is compared and it is overlapped. Okay, so one step is done. After that, the log FC values are also compared. Okay, so as I have told you that the log FC values, the expression values in the normal individual are 1, but here in this case, in the cancer patient, it is 5, 4.53, and 2.9. So it means that the ABCD genes are highly expressed. So what does this mean? It means that first the glycolysis pathway is enriched because all the genes are present, and after that it also have highly log FC values. Okay, so just because of that highly expression, the glycolysis pathway is enriched. So this means that the glycolysis pathway gene set is enriched and glycolysis pathway is highly regulated. Okay, so glycolysis pathway genes are overlapped plus also highly expressed. So we can say that glycolysis pathway is highly expressed in cancer patients. Okay, so I have just shown you a very simple glycolysis pathway. The genes are expressed and overlapped. So basically, the basic principle of gene set enrichment analysis is that we want to see the pathways. We want to see that our genes, which our genes are. For example, our input genes are how they are overlapping with different other genes, and after that, the log FC values tells us that our genes are overlapped, but if they are highly expressed, so which pathways are pathways are included. Okay, so this was a brief overview about the gene set enrichment analysis. Now coming to our very basic topic, that how does the input file will look like? So basically, this is our rank list. It consists of 2 columns: the symbol, gene symbol, and the log FC value. Okay, I have told you, and the rows are the genes and the expression, the log FC. Basically, if you are preparing a file, so it should consist of these 2 columns. Okay, and it should consist of symbols because symbols are highly accepted and normally accepted by the GSEA database. And if it consists, if you are having probe IDs, so then it will just there will be a problem because it does not like the probe IDs because it cannot interpret it. Okay, so if you're having the gene symbols and with specific log FC values, so then it is good enough for you that you can run a smooth GSE analysis. Okay. Okay, so this is the input file. As I have told you, these are the gene symbols and these are the log FC values of the gene symbols. So your input file should look like this. How does this file come? Where does it come from? So basically, it comes from the differential gene analysis. If you are processing your microarray data or you are processing your RNA-seq data, so in the last you can get this type of table. Okay, So in that table, you have your gene samples and your log FC value. Okay, and you know that what type of data it is, if it is cancer patient or if it is blood disease. So then you can easily relate your result with the GSEA database. Okay, so you can also through coding you can just remove these columns and you can interpret it, or if you can also manually just copy these and make another file, and after that paste it in, like this, and after that upload this file into the RStudio. Okay, so this will be another topic for the next video, that how you will upload the files and then how you will make it ready for the GSE analysis. But before it, you should know about your rank list, your input file. So this was all about today's video. I have told you that how to what is basically GSE analysis and how to prepare data file for GSE analysis. Okay. If you have any questions, just ask me in the comment box, or you can also email me. I will answer you there. Okay. So stay tuned for the next video. Thank you. God bless.